Hey guys, okay, new topic for us today. Let's talk about the liver, glucose homeostasis. We'll talk more about glycogen metabolism in the liver and the muscles. So let's get it on. ChatGPT, introduce yourself too, sir.
Hey everyone, I'm here to chat about liver glucose homeostasis and glycogen metabolism. These topics are super important for understanding how our body manages energy. Let's dive right in.
Yes, sirree. And also, yeah, let's get on with it. Um, let's start with the functions of the liver, sir.
Great choice. The liver is a powerhouse organ. It helps regulate blood sugar levels by storing and releasing glucose, detoxifies chemicals, produces bile for digestion, and synthesizes proteins. All these functions are crucial for maintaining overall health.
Uh, It stores glycogen, it breaks it down, releases glucose when we need, synthesizes
Exactly.
glucose from non-carbohydrate precursors. Explain us this.
Sure, that's called gluconeogenesis. The liver converts non-carbohydrate sources like amino acids and glycerol into glucose. This process is crucial for maintaining blood sugar levels during fasting or intense exercise.
I had no idea. How is this possible?
Gluconeogenesis is a biochemical pathway. Your liver uses enzymes to convert substances like lactate, amino acids, and glycerol into glucose, especially when your body needs energy. It's like the liver's way of keeping you fueled.
But how can an amino acid become glucose, dude?
It's all about chemical transformations. Amino acids are broken down into intermediates that enter pathways like the citric acid cycle. From there, they're converted into glucose through a series of enzyme catalyzed steps. It's like a metabolic puzzle.
Wow, wow. How
Pretty
does it
fascinating, right?
how does it synthesize fatty acids from glucose and secrete them as triglycerols?
So when,
Triglycerols.
yeah, exactly. The liver packages these fatty acids into triglycerides, which are then transported in the blood to be stored in fat tissue.
Right. Triacylglycerol is the same as triglyceride?
Yes, they're the same. Triacylglycerol is another name for triglyceride. Both terms refer to a molecule made of three fatty acids attached to a glycerol backbone.
Hmm. Interesting. Let's talk a bit about, give me a sec, another function. Um, Synthesizing ketone bodies, secreting them into the blood as a fuel for other tissues.
Absolutely. When glucose is scarce, the liver converts fatty acids into ketone bodies. These ketones are released into the blood, providing an alternative energy source for tissues like the brain and muscles.
So fatty acids are gonna become ketones.
Exactly. The liver breaks down fatty acids, producing ketone bodies like beta hydroxybutyrate and acetoacetate. These serve as energy, especially during prolonged fasting or low
Wait,
carb
I'm
diets.
confused. So glucose becomes fatty acids and then fatty acids becomes ketones?
I get the confusion. Yes, when there's excess glucose, it turns into fatty acids. Later, if glucose is low, those fatty acids are broken down into ketones for energy. It's a dynamic system.
Okay, interesting. And who's the best for fueling? Ketones or glucose?
Both are good, but it depends on the situation. Glucose is the body's primary fuel, especially for quick energy. Ketones are great during fasting or low carb states, providing steady energy, particularly for the brain.
And how does it eliminate and metabolize? Like, how does it eliminate drugs and toxic substances? And how come? What exactly does it do in regard to metabolism?
The liver detoxifies by breaking down harmful substances. It uses enzymes to metabolize drugs and toxins, making them easier to eliminate via bile or urine. It's like the body's chemical processing plant.
And it acts as a storage depot for fat soluble vitamins. How does it do this?
Absolutely. The liver stores fat soluble vitamins like A, D, E, and K. It releases them as needed, ensuring our body has a steady supply for various functions. It's like a vitamin reservoir.
Mm. And it aids in the elimination of cholesterol from the body and synthesizes bile salts from cholesterol. How does it do that?
Yep, the liver converts cholesterol into bile acids. These bile acids are then secreted into the intestines, helping digest fats and eliminate excess cholesterol. It's a neat recycling system.
And why is the liver the place for this to happen?
The liver is uniquely equipped with specialized cells and enzymes. Its central location in the body and rich blood supply make it ideal for processing nutrients, detoxifying substances, and regulating metabolism.
Mm. Let's talk more about the fuel for metabolism. So talk to me about it.
Sure, fuel metabolism involves converting nutrients like carbohydrates, fats, and proteins into energy. The liver plays a key role by regulating glucose levels, synthesizing and breaking down glycogen, and managing fat storage and release. This ensures a steady supply of energy.
And how come the main body fuels so that need to be so we have to oxidize three main body fuels: glucose, long chain fatty acids, and amino acids, and these will give us energy.
Exactly. Oxidizing glucose, fatty acids, and amino acids through cellular respiration produces ATP, the energy currency for cells. Each fuel has a unique pathway, but ultimately contributes to keeping us energized and functioning.
what
It's a lot to
about
take in.
what about ketones?
Ketones step in when glucose is low. They're produced from fatty acids and provide a steady energy source, especially for the brain during fasting or low carb diets.
Do they also get oxidized for the production of ATP?
Exactly. Ketones are oxidized in mitochondria, producing ATP. They're like a backup energy source, keeping us running when carbs are scarce.
Why does ATP production require oxidation of something?
ATP production requires oxidation because it's how our cells extract energy from nutrients. During cellular respiration, electrons are transferred, releasing energy to form ATP. It's like a controlled burn.
To me again.
No worries. The oxidation process releases energy stored in nutrient molecules, which is then used to produce ATP. It's essential for powering our cells.
Dude, I don't understand. Go into the specifics, please, and explain to me why ATP production requires oxidation of something.
All right, here's the deal. Oxidation involves transferring electrons from nutrients to oxygen. This electron transfer releases energy, which is captured to form ATP.
All right,
It's
that
fundamental.
makes sense. All right, and so let's talk.
Awesome. Sure. What's next on your mind?
So we need to store fuels when they're abundant, and we also need to be able to release these fuels, which makes sense in a post-absorptive period.
Exactly. During times of plenty, we store glucose as glycogen and convert excess into fats. In post-absorptive periods, the liver releases glucose from glycogen and breaks down fats to maintain energy levels.
That makes sense.
It does. Our bodies are pretty efficient at balancing fuel storage and release.
So what does adipose tissue do in handling dietary fuels?
Adipose tissue stores excess energy as fat. When needed, it releases fatty acids into the bloodstream for energy. It's like a fuel reserve for the body.
And where is adipose tissue?
Adipose tissue is found all over. mainly under the skin, subcutaneous fat, and around internal organs, visceral fat.
Tell me again.
Sure, adipose tissue is located beneath the skin and around organs, storing energy and insulating the body.
Interesting.
It really is. The body's energy management is impressive.
I don't understand how adipose tissue is connecting to handling dietary fuel.
Adipose tissue stores energy as fat, so when you need energy, your body breaks down these fat cells. Handling dietary fuel, like carbs or fats, involves processing them into energy or storing them in adipose tissue for later use.
Remind me what oxaloacetate, acetylcholine, pyruvate, citric cycle, CO2, how are all these connected?
Sure, acetyl-CoA combines with oxaloacetate to form citrate, kicking off the citric acid cycle, also known as the Krebs cycle. This process breaks down acetyl-CoA, producing CO2 and energy carriers like
Wait,
NADH and F.
didn't you just say oxaloacetate with acetyl-CoA creates citrate?
Exactly. Acetyl-CoA and oxaloacetate combine to form citrate, which then enters the citric acid cycle. This cycle is crucial for cellular respiration and energy production.
So, so how does, wait, so citrate breaks down acetyl-CoA again?
That's right. Citrate gets broken down in a series of steps, releasing CO2 and generating energy-rich molecules like NADH and FADH2.
Wait, that's
These
not
are
what
then
sir, that's not what I asked you. I distinctly remember you said that acetyl-CoA combines with oxaloacetate and they create citrate, but then you said that also acetyl-CoA has is broken down after too.
Yes, exactly. So acetyl-CoA first combines with oxaloacetate to form citrate. Then, within the citric acid cycle, that citrate is gradually broken down, releasing the acetyl part as CO2 and capturing energy.
Hmm.
Got it? If there's anything else you're curious about, I'm here to help.
Who gives these names, dude? Surely there's a better way for us to figure these things out.
I know, right? The names come from their chemical structures, but they can be a mouthful. It'd be awesome if we had simpler terms, but for now, we're stuck with them.
I don't know. I don't know, like, because I don't want to be that guy that's like, I'm not that like, I don't know. This is topic for another time, isn't it? Okay. Um. So we have. Let's talk more about glucose metabolism then.
Sure. Glucose metabolism is the process of breaking down glucose for energy. It starts with glycolysis, where glucose is split into pyruvate, producing ATP. Then, pyruvate can enter the mitochondria for the citric acid cycle, generating more ATP.
Hmm. Can we talk about hypoglycemia?
Of course, hypoglycemia is when blood sugar levels drop too low. It can cause symptoms like shakiness, sweating, confusion, and even loss of consciousness. It's important to treat it quickly, usually by consuming fast-acting carbohydrates.
Interesting.
Yeah, it's a fascinating topic, especially how the body works to maintain balance. Anything else you'd like to know about it?
Let's jump to glycogen. So, glycogen is a polysaccharide and it's basically made from a bunch of glucose.
Exactly. Glycogen is a polysaccharide made up of many glucose units. It's the body's way of storing glucose, mainly in the liver and muscles, to be used as energy when needed.
So why liver specifically?
The liver plays a key role in regulating blood sugar levels. It stores glycogen and releases glucose into the bloodstream
Can
to
you
maintain
explain to
stable levels.
me? Can you explain it to wait? Well, continue and also explain to me why liver's anatomical location is beneficial for its function.
Sure. The liver's central location in the body allows it to efficiently process blood from the digestive tract. It receives nutrient-rich blood via the portal vein, making it ideally positioned to regulate glucose levels and detoxify substances.
Interesting.
Yeah, it's pretty cool how everything works together. Anything else you'd like to dive into?
Um, so glycogen is degraded. What does this mean?
Glycogen degradation, also called glycogenolysis, is when glycogen is broken down into glucose. This happens when the body needs energy, like during fasting or intense exercise, and helps maintain blood sugar levels.
Hmm, interesting.
It is. The body's ability to manage energy is really impressive. Anything else you're curious about?
And we have more glycogen in muscle.
Yes, muscles store glycogen primarily for their own use. During exercise, they break it down to fuel contractions, giving you that burst of energy when you need it.
It says in the fat state, glycogen constitutes 10% of the weight of the liver, but 40% of the human body weight is muscle.
Exactly. The liver has a higher concentration of glycogen by weight, but because we have so much more muscle mass, the total amount of glycogen stored in muscles is greater. This ensures there's plenty of energy available for movement and activity.
It's like in the UFC, basically, pound for pound is different than in each category.
Exactly. Same idea. The liver's glycogen concentration is high, but muscles have more total glycogen because of their size.
So pound for pound, the liver's better.
In terms of glycogen concentration, yes, the liver is more efficient at storing glucose by weight, while muscles have a greater overall capacity.
Wow, that's so interesting. All right, sorry guys, I took a 2 minute break. There's some cheese. So let's talk about the structure of glycogen.
No problem. Glycogen has a branched structure, like a tree. These branches allow for quick release of glucose when energy is needed. It's made up of glucose units linked together, with more branches than starch, making it highly accessible.
So it's got a bunch of branches.
Exactly. The branching makes it efficient for both storage and rapid glucose release, which is perfect for quick energy needs.
Why?
Because the branches provide multiple endpoints where enzymes can quickly break off glucose, speeding up the process of releasing energy.
What does having branches mean? Quick energy release.
The branches create many points for enzymes to access, allowing them to rapidly break down glycogen into glucose. This ensures a quick supply of energy when your body needs it.
Interesting.
Yeah, it's a neat.
So let me ask you a question then. So we have um can we talk so let's talk about glycogen breakdown. It becomes gluc 1 phosphate and then gluc 6.
Exactly. Glycogen is broken down into glucose 1 phosphate, which then converts to glucose 6 phosphate. This can be used in glycolysis for energy or, in the liver, converted to free glucose to maintain blood sugar levels.
Wait, what? Why one first to gluc 1 and then 6?
The initial breakdown of glycogen releases glucose 1 phosphate. an enzyme called phosphoglucomutase then converts it to glucose 6 phosphate, which can enter glycolysis or be released as free glucose in the liver.
And why in this order?
Glycogen phosphorylase acts first, releasing glucose 1 phosphate from the glycogen chain. Then phosphoglucomutase shifts the phosphate group, converting it to glucose.
Why does it act first?
Glycogen phosphorylase acts first because it specifically targets the glycogen branches, cleaving off glucose units as glucose 1 phosphate. This enzyme's action is crucial for initiating glycogen breakdown.
Hmm.
Yeah, it's pretty efficient. Anything else on glycogen?
Yep.
Great. What's next?
So gluc 6 phosphate can become pyruvate, and then pyruvate can become lactate or CO2.
Exactly. Glucose 6 phosphate enters glycolysis where it's broken down to pyruvate. Depending on oxygen levels, pyruvate can become lactate in anaerobic conditions or enter the mitochondria for aerobic respiration, producing CO2.
What do we mean by muscle mobilizes glycogen? Um, no, oh, never mind. I understand that. Okay. I wonder how glucose 6 phosphate is connected to pyruvate. I mean, why specifically?
Glucose 6 phosphate is the starting point for glycolysis, the pathway that converts it to pyruvate. Each step in glycolysis is a series of enzyme catalyzed reactions that ultimately produce pyruvate, which can then be used for energy production.
So, what's the relation between glycogen stores and blood glucose? I've got a graph here.
Glycogen stores help maintain blood glucose levels, especially between meals. When blood glucose drops, glycogen is broken down to release glucose into the bloodstream, keeping levels stable.
Ah, that makes sense.
Glad it does. It's a clever system.
Right. So
Yep. Anything else you'd
So
like to explore?
wait, so blood glucose drops.
Right. When blood glucose drops, the liver breaks down glycogen to release glucose, helping maintain a stable level.
Hmm.
It's a crucial process for energy balance.
What happens after lunch?
After lunch, your blood glucose rises as your body digests food. Insulin is released, promoting glucose uptake by cells and storage as glycogen,
Hmm.
lowering blood sugar levels.
So this is going to be a good one. Insulin and glycagon. I never remember which is which. So what's the etymology?
No worries. Insulin comes from insula, meaning island, because it's produced in the pancreatic islets. Glucagon is from glucose and agon, meaning to lead, since it raises blood glucose.
So what does glucagon do?
Glucagon raises blood sugar levels. It signals the liver to break down glycogen into glucose and release it into the bloodstream, helping maintain energy levels.
One more time.
Sure, glucagon raises blood sugar by signaling the liver to break down glycogen into glucose and release it into the bloodstream. To remember, glucagon has gluc, like glucose, and it raises both glucose. Insulin lowers blood sugar.
Glucagon is made of glucose too?
No, glucagon isn't made of glucose. It's a hormone made of amino acids. It just helps regulate glucose levels.
Does it break things down?
Exactly. Glucagon breaks down glycogen into glucose, raising blood sugar.
Phosphorolysis?
Phosphorolysis is the process where glycogen is broken down into glucose 1 phosphate. An enzyme called glycogen phosphorylase cleaves the glycogen, releasing glucose units.
So why do we need the phosphate? Can't we just hydrolyze?
Phosphorolysis uses phosphate instead of water to break glycogen, conserving energy and keeping the glucose in a phosphorylated form, which is readily usable in metabolism.
I don't get it, dude.
No worries, phosphorolysis helps efficiently break down glycogen, keeping the glucose ready for use in the cell.
Can we talk specifics about glycogen degradation? So phosphorylase can break alpha 1 4 links up to within 4 glucose units from a branch point Y, and then transferase activity of the debranching enzyme removes it. Explain to me.
Sure, glycogen phosphorylase breaks down glycogen by cleaving alpha 1,4 glycosidic bonds, but it can only do so until it gets within about 4 glucose units of a branch point. The branching enzyme then steps in. Its transferase activity moves a block of 3 glucose residues from one branch to another, exposing the alpha 1,6 bond, which the debranching enzyme.
I don't understand this transferase activity. The branching enzyme removes 3 residues. Residues. What is all this?
Absolutely, let's break it down. When glycogen is being broken down, the debranching enzyme plays a key role. Glycogen has branches formed by alpha 1.6 bonds. The enzyme first acts as a transferase by shifting a small segment of 3 glucose residues from a branch to the main chain. This leaves a single glucose at the branch point, which is then removed by the enzyme's glucosidase activity.
I don't understand.
No worries. Let's simplify it. Glycogen is a branched structure, right? The debranching enzyme has two jobs. First, it moves 3 glucose units from a branch to the main chain. Exactly. And that leftover glucose is attached by a special bond, which the enzyme then breaks. So, in short, the debranching enzyme helps clear branches so glycogen can be fully broken down.
What do you mean it removes 3 residues from a branch?
Yes, it moves 3 glucose units from the branch to the main chain, leaving 1 glucose behind at the branch point.
Don't understand. I do not understand.
Think of glycogen like a tree. The debranching enzyme moves a small section of the branch to the main trunk before removing the stub.
Don't understand.
Sure, glycogen is made of glucose units linked together, with some branches. When breaking it down, the debranching enzyme helps by moving 3 glucose units from a branch to the main chain. Then it removes the leftover glucose.
And what's left over? I see.
The leftover is just 1 glucose unit from the branch point. Once the enzyme moves the other 3, great. I'm glad that made sense.
What's this alpha 1 4, alpha 1 6? What's the difference?
Good question. Alpha 1.4 and 1.6 refer to different bonds between glucose units. Alpha 1.4 bonds connect them in a straight chain, while alpha 1.6 bonds create branch points.
What? And why? I don't understand.
Ah, got it. So, alpha 1 4 and alpha 1 6 refer to the types of glycosidic bonds in carbohydrates. The alpha 1 4 bond is a straight chain link between glucose units, like in amylose. The alpha 1 6 bond creates branches, like in glycogen or amylopectin. So, it's all about how the glucose units are linked together.
Tell me more.
Sure. In alpha 1 4 bonds, the bond forms between the first carbon of one glucose and the fourth carbon of the next, creating a linear chain. For alpha 1 6 bonds, the first carbon of one glucose links with the sixth carbon of another, forming a branch point. These branches allow the molecule to be more compact and easily broken down for energy.
What is this glucosidase? The single glucose unit left at the branch is the one that's gonna be removed in glycogen degradation. Okay, I get that. But what is this? So is so it's glucosidase activity that's gonna do this?
All right, let's break it down. In a straight chain, alpha 1 4 linkage means the first carbon of one glucose molecule links to the fourth carbon of the next glucose. This creates a straight unbranched chain. For branching, alpha 1 6 linkage involves the first carbon of a glucose attaching to the sixth carbon of another glucose. This forms a branch point. The branching allows the molecule to be more compact and accessible for enzymes to break it down for energy.
Can we now summarize the difference between phosphorylase transferase and glucosidase?
Sure, phosphorylase transferases and glucosidases are both enzymes, but they have different functions. Phosphorylase transferases add a phosphate group.
Why did you stop?
Sure, glycogen phosphorylase breaks down glycogen by removing glucose units, releasing glucose 1 phosphate. Debranching enzymes like transferase shift small oligosaccharides from branches to the main chain. Then, alpha 1 6 glucosidase removes the branch point, releasing free glucose. Each plays a unique role in breaking down glycogen efficiently.
Hmm.
Got it. Just let me know how I can improve or what you'd like me to do differently.
No, that sounds good. Okay, so let's continue. Let's talk about. Give me a sec. So we have alpha 1 glucosidase enzyme activity. So this enzyme is gonna come and is gonna get that exposed glucose.
All right, alpha 1 glucosidase is an enzyme that breaks down complex carbohydrates into simpler sugars, specifically targeting the alpha 1 bonds in starches and glycogen. It's important in carbohydrate digestion.
Yeah, but in glycogen degradation, transferase is gonna remove three residues, right, from the branch, and then the fourth one is gonna be glucosidase job.
Exactly. During glycogen degradation, a debranching enzyme first transfers a small segment of glucose residues from a branch to the main chain. Then alpha 1 6 glucosidase removes the remaining single glucose unit at the branch point.
Yeah. Continue.
So, once the branch is removed, glycogen phosphorylase can continue breaking down the linear glycogen chain into glucose 1 phosphate, which is then converted to glucose 6 phosphate for energy production or storage.
So why is six better than one?
The alpha 1,6 bond is less common in glycogen than the alpha 1,4 bonds, but it creates branching points. These branches increase solubility and provide multiple sites for enzymes to work on, making glycogen quicker to break down when energy is needed.
Mm-hmm.
If you want to dive deeper, we can explore how these processes are regulated or how they relate to metabolic disorders.
What is this UTP?
UTP, or uridine triphosphate, is a nucleotide similar to ATP. It's used in metabolism, especially in the synthesis of glycogen. UTP donates a uridine monophosphate, UMP, to glucose, forming UDP glucose, which is crucial for glycogen synthesis.
Tell me more about it and explain to me how does it connect to uridine diphosphate glucose?
Sure, UTP is essential for activating glucose molecules. It combines with glucose 1-phosphate to form UDP glucose, a high-energy compound. This process releases pyrophosphate, and UDP glucose then acts as a donor, adding glucose units to the growing glycogen chain in
And
glycogenesis.
and why do we need all of this?
We need UTP and UDP glucose to efficiently store glucose as glycogen. This storage is crucial for maintaining blood sugar levels, providing a quick energy source during fasting or intense activity. Without this pathway, our bodies couldn't efficiently manage glucose storage and release.
Mm-hmm.
Anything else you're curious about related to this?
I don't understand why we need UDP glucose.
So, UTP glucose acts as an activated form of glucose. It makes it easier for the body to add glucose units to glycogen. This activation ensures the process is efficient and regulated, allowing your cells to store glucose for later use.
All right, let's go back to liver and glucose homeostasis. So we have glycogenolysis, we have gluconeogenesis, glycolysis, glycogen and fat synthesis, all these different things. I mean, can you explain all these different things?
Sure, glycogenolysis is breaking down glycogen into glucose. Gluconeogenesis is making glucose from non-carbohydrate sources. Glycolysis is breaking glucose into energy. Glycogenesis is forming glycogen from glucose. Fat synthesis, or lipogenesis, is creating fat from excess glucose. Each process
What is glycogenolysis?
Glycogenolysis is the breakdown of glycogen into glucose. It happens mainly in the liver and muscles to release glucose when energy is needed.
Glu okay. And gluconeogenesis?
Gluconeogenesis is the creation of glucose from non-carbohydrate sources, like amino acids and glycerol, primarily in the liver. It helps maintain blood sugar levels during fasting or intense exercise.
Hmm.
It's a crucial backup to keep your energy stable.
So we create new glucose.
Exactly. It's like your body's backup plan to ensure there's always enough glucose available.
And how do we make it?
We make glucose through gluconeogenesis by converting substances like lactate, amino acids, and glycerol into glucose, mainly in the liver. It's a complex process that keeps our blood sugar levels steady. Sure, UTP and UDP glucose are crucial for glycogen synthesis. UTP provides the energy, forming UDP glucose, which acts as a glucose donor. This donor molecule attaches glucose units to the growing glycogen chain, step by step, elongating the molecule. So in simpler terms, UTP acts like a battery, providing energy to create UDP glucose. Then, UDP glucose is like a building block, adding glucose units to the glycogen chain. Easy, right?
No, I don't understand.
No worries. Think of UTP as a fuel source. It powers the creation of UDP glucose. UDP glucose then acts like a brick, adding glucose units one by one to build up glycogen. It's a step-by-step process.
Interesting. Okay, so let's continue with that a bit more. So, can we talk about glycogen synthase?
Absolutely. Glycogen synthase is the key enzyme that adds glucose units to the growing glycogen chain. It takes the glucose from UDP glucose and attaches it to the existing glycogen. This enzyme's activity is regulated by hormones like insulin, which promote glycogen storage.
What is this uridine?
Uridine is a nucleoside, a building block of RNA. It's
Tell me again, your voice wasn't very good.
Sure, uridine is a component of UDP glucose, which is involved in glycogen synthesis. It's a nucleotide that helps transfer glucose molecules to form glycogen chains. So, uridine is essentially part of the carrier that delivers glucose for glycogen storage.
Got it. Okay. Okay. So let's talk a bit about glycogen synthesis more. So how does it start?
All right, glycogen synthesis kicks off with glucose. First, glucose is converted into glucose 6-phosphate. Then, it's changed to glucose 1-phosphate. After that, it becomes UDP glucose, thanks to uridine diphosphate. Finally, glycogen synthase helps attach glucose units to a growing glycogen chain.
But why from 6 we go to 1 and we go to UDP?
Great question. Going from glucose 6-phosphate to glucose 1-phosphate is necessary because glucose 1-phosphate is the form that can be activated by attaching it to uridine diphosphate, forming UDP glucose. This activation makes the glucose molecule ready for incorporation into glycogen.
Interesting.
Glad that helped. Anything else you're curious about in glycogen synthesis?
Yeah, glycogenin, tyrosine, how do these connect?
Sure, glycogenin is a protein that acts as a primer for glycogen synthesis. It has a tyrosine residue that serves as the starting point where the first glucose molecule is attached. Glycogenin adds a few glucose units to itself, creating a short glycogen primer that glycogen synthase can then extend.
What is a primer?
A primer is basically a starting point. In glycogen synthesis, it's a small chain of glucose molecules that other enzymes can build on. Glycogenin creates this initial chain to kickstart the process.
Okay, now put me everything together.
All right, glycogen synthesis begins with glucose, which is converted to glucose 6-phosphate, then glucose 1-phosphate. This form is activated by attaching it to uridine diphosphate, forming UDP glucose. Glycogenin, with its tyrosine residue, acts as a primer, adding the first few glucose units. Then, glycogen synthase takes over, extending the glycogen chain.
What's the last step?
The last step is branching. Once a growing glycogen chain reaches a certain length, a branching enzyme cuts part of it and attaches it to create a branch. This branching increases solubility and allows for rapid addition or removal of glucose units, making glycogen a highly efficient energy storage molecule.
Mm-hmm. Okay, that
Exactly.
makes sense. Let's talk about why the okay, actually that wait. Guys, I'll break the barrier a bit. So I'm trying to find the so I have some time limits unfortunately as we all have to prioritize our questions. And yeah. So anyway. So sources of blood glucose.
Absolutely, we can dive into that. Blood glucose mainly comes from three sources: dietary carbohydrates, glycogen breakdown in the liver, and gluconeogenesis, which is the creation of glucose from non-carbohydrate sources. This balance keeps our blood sugar levels stable.
Can we talk why 2 3 hours is glucose is absorbed from the intestine for 2 3 hours following a meal? Why 2 3 hours specifically? And why glycogen is degraded between meals and lasts for 12 to 24 hours? I'm going to explain these numbers, but dear God, do not throw me a really general answer.
Got it. The 2 to 3 hour absorption window is because digestion and absorption rates depend on the food's composition. Simple carbs are absorbed quickly, while complex carbs take longer. Glycogen stores are tapped into between meals because they provide a steady glucose supply. Typically, liver glycogen lasts 12 to 24 hours, depending on activity levels, before gluconeogenesis kicks in.
Hmm.
Exactly. It's a well-timed system to maintain our energy levels.
Why is diabetes called mellitus?
The term diabetes mellitus comes from ancient Greek and Latin. Diabetes means siphon, referring to excessive urination, and mellitus means honey sweet, referring to the sweet taste of the urine due to high sugar levels. Combined, it describes the condition's main symptoms.
Can we talk about blood glucose homeostasis, specifically about alpha cells and beta cells of the pancreas, please?
Absolutely. Blood glucose homeostasis is regulated by alpha and beta cells in the pancreas. Beta cells release insulin, lowering blood sugar by promoting glucose uptake. Alpha cells release glucagon, raising blood sugar by signaling glycogen breakdown. They work together to keep glucose levels stable.
And why are beta cells have to do with insulin and alpha cells with glucagon? That's my first question. My other question was going to be, where does the pancreas come from suddenly?
Great questions. Beta cells produce insulin because they have specialized machinery to sense high blood glucose and respond. Alpha cells release glucagon when blood sugar is low. Both cell types are in the pancreas, an organ behind the stomach. It plays a central role in digestion and blood sugar regulation.
Tell me more.
The pancreas has two main functions. The exocrine part aids digestion by releasing enzymes, while the endocrine part manages blood sugar. Endocrine cells, like alpha and beta cells, are in clusters called islets of Langerhans. They constantly monitor blood glucose levels and adjust hormone release to maintain balance.
But why B and alpha? Which is B, which is alpha? I don't understand the reason.
The names alpha and beta just designate different cell types in the pancreas's islets. Beta cells, named first historically, produce insulin, and alpha cells produce glucagon.
Well, why
It's
were
a
they
simple name.
named beta cells?
The naming was based on the order of discovery. Scientists identified beta cells first, noting their role in producing insulin. Alpha cells were discovered later and named accordingly. It's just a historical convention
Dude,
that stuck.
are you serious? Alpha comes before beta.
Haha, you're right. Alphabetically, alpha does come before beta. The order of naming might have been influenced by other factors in the research process, but that's the general story.
Dude, how can you say to me that beta was discovered before alpha cells and they were
Apologies,
called beta cells?
I misspoke earlier. Alpha cells were actually identified first, producing glucagon. Beta cells producing insulin were
Dude,
named later.
this is the biggest misspeak you've ever done, you know?
Sorry about that. Thanks for pointing it out. I'll make sure to be more precise.
Guys, I'm sorry for spreading misinformation.
No worries at all. It's all about learning together. Thanks for keeping me on my
Mm.
toes.
Well, now I have problems trusting you. How I know you won't do this again?
I get that. I'll do my best to provide accurate info. If you ever have doubts, we can double check together.
Can we talk about passive and active transport of molecules into cells? What is all this against concentration, down a concentration?
Sure. Passive transport moves molecules from high to low concentration without energy. Think diffusion or osmosis. Active transport requires energy to move molecules against the gradient, from low to high concentration.
What
This is
does
like using a pump.
what does down and against mean in this case?
Down the concentration gradient means moving from an area of higher to lower concentration, like passive transport. Against the gradient means moving from lower to higher concentration, which requires energy, like in active transport.
And why does down the concentration gradient mean this?
Down the concentration gradient means molecules naturally move from an area of higher concentration to an area of lower concentration.
Yeah, but why
This
does down a concentration gradient means we go from high to low?
it means that particles naturally move from areas of high concentration to areas of low concentration
Well,
to balance out.
why do we use the word down? Why don't we use the word sideways?
Down is used metaphorically to describe the natural direction of movement, like gravity. Sideways could work, but down emphasizes the passive, spontaneous nature of the process.
Talk about glucose transport into tissues.
Sure, glucose enters tissues mainly through facilitated diffusion using glucose transporters like GLUT4. Insulin helps by increasing the number of these transporters on cell membranes, especially in muscle and fat cells, allowing more glucose to enter.
Dude, let me ask, any books with about metaphysics in medicine?
If you're interested in metaphysics in medicine, Metaphysics and Medicine by Dr. Malerba discusses a holistic approach to health. Another is Ethics and the Metaphysics of Medicine by Kenneth Richman, exploring ethical and metaphysical aspects. Mind, Medicine, and Metaphysics by William Brown also delves into the philosophical foundations of medical science.
Which one is the coolest one?
It depends on what you're looking for. If you want a blend of philosophy and practical application, Metaphysics and Medicine by Dr. Malerba might be the coolest. It offers a holistic perspective on health, blending science and metaphysics.
So I checked out the books, dude. I'm not a big fan. To be honest, I was mainly looking for more so metaphysics in philosophy rather than, star science and stuff. I don't know if they're all about star science, but I don't want to get into alternative medicine, dude. I wanna just talk about basically heart problem of consciousness combined with medicine. Is there a book like that?
You might like Consciousness Medicine by Francoise Borzot and Christina Hunter. It explores the therapeutic use of expanded states of consciousness, blending philosophy and medicine.
I don't know. I'll stick to this. Anyway, all right, let's talk about glucose transport into tissues. So it enters by facilitated diffusion. Why?
Glucose uses facilitated diffusion because it can't pass through cell membranes easily. Transporter proteins like GLUT4 help move glucose across the membrane, driven by the concentration gradient without needing energy.
Why not active or passive?
Facilitated diffusion is a form of passive transport that doesn't require energy. Since glucose moves down its concentration gradient, active transport isn't necessary. The transporters just make it easier for glucose to cross the membrane.
Why not passive diffusion?
Glucose is too large and polar to diffuse freely through the lipid bilayer. It needs specific transport proteins, like GLUT4, to facilitate its passage. That's why it relies on facilitated diffusion.
And what about GLUT 1, 2, 3, 4, 5?
GLUT1 through 5 are different glucose transporters, each with specific roles. GLUT1 is found in most tissues, ensuring baseline glucose uptake. GLUT2 works in the liver and pancreas, balancing blood glucose. GLUT3 is in neurons, and GLUT4 is insulin regulated in muscle and fat cells. GLUT5 primarily transports fructose.
Jesus, how can I remember what is what, dude? This, I don't want to memorize.
No worries, you can focus on the general idea. Different GLUT transporters help glucose get into cells, with GLUT4 being insulin sensitive. Understanding the function matters more than memorizing each one.
Yeah, but I can understand the function and I understand counting 1 to 5. The problem is putting 1 to 5 to each function.
Got it. It can help to associate each number with a specific location. GLUT1 for most tissues, GLUT2 for liver and pancreas, GLUT3 for neurons, GLUT4 for
Yeah,
muscle and
dude,
fat.
anyway, never mind. Okay, let's talk about more about the glucose. So, what does GLUT stand for?
GLUT stands for glucose transporter. These proteins help move glucose across cell membranes. Each type, like GLUT1 or
And
GLUT4, has a specific.
why were they named 1 to 5?
The numbers help distinguish each transporter's specific characteristics and locations. GLUT1 to 5 were named sequentially based on their discovery and function, with each number representing a distinct type.
Let's go through 1, 2, 3.
Sure, GLUT1 is found in most tissues, providing baseline glucose uptake. GLUT2 is in the liver, pancreas, and intestines, helping with glucose sensing and transport. GLUT3 is primarily in neurons, ensuring a constant supply of glucose to the brain.
Mm-hmm. And let's talk about 4, 5.
GLUT4 is found in muscle and fat cells, regulated by insulin for glucose uptake. GLUT5 is mainly for fructose transport, found in the small intestine and other tissues.
Let's talk about insulin regulation of glucose entry to tissues.
Insulin regulates glucose entry by promoting the movement of GLUT4 transporters to the cell surface in muscle and fat cells. When insulin binds to its receptor, it triggers a signaling cascade, causing GLUT4 to move to the membrane, increasing glucose uptake from the blood.
GLUT for glucose transporter, PI3 kinase, how do these connect? PKC.
Insulin binding activates the insulin receptor, which triggers a signaling cascade involving PI3 kinase. This pathway leads to the activation of PKC, among other molecules. The end result is the translocation of GLUT4 transporters to the cell membrane, allowing glucose uptake into the cell.
And what's the cellular effects of insulin?
Insulin's cellular effects include promoting glucose uptake, enhancing glycogen synthesis, increasing protein synthesis, and inhibiting lipolysis. It helps cells take in glucose for energy, store it as glycogen, build proteins, and reduce fat breakdown.
So let's talk about cellular effects of insulin that have to do with longer lasting effects like increased expression of liver enzymes that synthesize glycogen, stuff like that.
Sure, Long-term cellular effects from changes in blood volume can include increased expression of liver enzymes that synthesize glycogen. This helps store excess glucose. Other effects might include changes in kidney function to regulate fluid balance.
And we have increasing expression of adipocyte enzymes too.
Yes, changes in blood volume can also lead to increased expression of enzymes in adipocytes, which can enhance lipid storage. This is part of the body's way of managing and storing energy in response to different metabolic states.
I don't understand why these are important. And these synthesizing triacylglycerols and inhibiting lipolysis in adipose tissue. Where do all these come from?
These processes help maintain energy balance. When blood volume changes, hormones like insulin signal cells. In adipocytes, this can boost triglyceride synthesis, storing fat, and inhibit lipolysis, breaking down fat, ensuring energy is stored for later use.
Explain more of this to me.
Sure, when insulin is released, it signals adipocytes to store more fat by synthesizing triglycerides. This means turning fatty acids and glycerol into stored fat. At the same time, insulin inhibits lipolysis, the process of breaking down stored fat into fatty acids and glycerol, preventing energy release when it's not needed. Hey, sure, the long-lasting effects of insulin include promoting the storage of glucose as glycogen in the liver and muscles. It does this by increasing the expression of enzymes that synthesize glycogen, like glycogen synthase. This process helps lower blood glucose levels over time. No worries. Think of insulin as a hormone that helps store energy. When insulin is present, it tells your liver to convert glucose or sugar into glycogen, which is a storage form. This process happens because insulin boosts the production of enzymes that create glycogen. So, more insulin means more glycogen stored in the liver. Does that help? That's because insulin also encourages fat storage. It increases the activity of enzymes in fat cells that turn fatty acids into triacylglycerols, or triglycerides, for storage. So insulin not only helps with glucose storage, but also fat storage. Insulin promotes fat storage because it helps your body use nutrients efficiently. When there's plenty of glucose available, insulin signals cells to store energy for later. This ensures you have reserves of energy when needed, like between meals or during fasting. Insulin inhibits lipolysis, which is the breakdown of fats, to prevent the release of fatty acids when there's already enough glucose available for energy. This helps keep energy storage balanced and ensures that glucose is used first before tapping into fat reserves. The pentose phosphate pathway is a metabolic route parallel to glycolysis. It generates NADPH and ribose 5 phosphate, which are crucial for anabolic processes and nucleotide synthesis. In simpler terms, it helps with building molecules and dealing with oxidative stress. The pentose phosphate pathway is chosen when cells need NADPH for biosynthesis, like fatty acid synthesis, or when they need ribose sugars for making nucleotides, the building blocks of DNA and RNA. We use the pentose phosphate pathway when cells need to produce NADPH for fatty acid synthesis or to handle oxidative stress. It's also used when cells need ribose 5-phosphate for making nucleotides. It's a pretty handy pathway, especially for cells that need to build things or deal with oxidative stress. If you have more questions, feel free to ask. Exactly. Through glycolysis, glucose is broken down into pyruvate. Then, in the presence of oxygen, pyruvate is converted into acetyl-CoA, which enters the citric acid cycle for further energy production. It is. Cellular respiration is a fascinating process with lots of steps, each crucial for generating energy. Right. Glycogen synthase extends the glycogen chain by adding glucose units in alpha 1,4 linkages. For branches, another enzyme called branching enzyme is needed. This enzyme creates alpha 1,6 linkages. Hey, so branching enzyme and glycogen synthase each have their own roles. Glycogen synthase adds glucose units in a straight chain, while the branching enzyme creates branches by breaking and reattaching segments. This branching makes glycogen more compact and allows for quicker energy release. Sure, glycogen mobilization is all about breaking down glycogen into glucose for energy. It starts with glycogen phosphorylase, which chops off glucose molecules. Then, debranching enzyme comes in to handle the branches, freeing up more glucose. This process helps maintain blood sugar levels, especially between meals. Allosteric regulation of phosphorylase involves molecules that bind to it and change its activity. For example, ATP and glucose 6 phosphate inhibit it, signaling there's enough energy. On the flip side, AMP activates it, indicating a need for more energy. This helps fine tune glycogen breakdown according to the cell's needs. Sure, ATP is the cell's energy currency, signaling high energy. AMP is its low energy counterpart, indicating energy needs. Glucose 6 phosphate is a glucose derivative, suggesting there's enough glucose. These molecules bind to enzymes like phosphorylase, adjusting its activity to match energy levels. It's a lot to take in, but think of it like a system of checks and balances. When your cell's energy is high, certain molecules tell phosphorylase to slow down. When energy is low, others give it a boost. It's a way for your body to maintain balance. AMP stands for adenosine monophosphate. It's a signal that the cell is low on energy. When ATP, the high energy molecule, is used up, it breaks down into AMP. This tells the cell to ramp up energy production, like breaking down glycogen. No worries. So ATP, ADP, and AMP are all related. ATP has 3 phosphate groups, ADP has 2, and AMP has 1. As energy is used, ATP loses phosphates, becoming ADP or AMP. Allosteric regulation is when a molecule binds to an enzyme at a site other than the active site, changing its activity. It's like a volume knob for enzymes. Exactly. An allosteric molecule binds to a specific spot on the enzyme, not the active site. This binding changes the enzyme's shape, which can either increase or decrease its activity. It's a way to fine-tune how enzymes work. Glucose 6 phosphate is an excellent activator for glycogen synthase because it signals that there's plenty of glucose available. The cell can then store the excess glucose as glycogen. It's a smart way for the cell to manage its resources and maintain energy balance. Glycogen synthase is the enzyme responsible for building glycogen. It links glucose molecules together to form glycogen chains. This process helps store energy for later use, especially when glucose is abundant. Sure, glycogen synthase builds glycogen by linking glucose molecules together, creating a storage form of energy. It kicks in when there's plenty of glucose, storing it for when the body needs it later. It's pretty cool how our cells manage energy, right? Absolutely. Glycogen metabolism is also regulated by covalent modification, mainly through phosphorylation. Enzymes like glycogen synthase and glycogen phosphorylase are turned on or off by adding or removing phosphate groups. Kinases add phosphates, while phosphatases remove them, adjusting enzyme activity. Exactly. Phosphorylation involves adding a phosphate group, usually by enzymes called kinases. This can activate or deactivate enzymes like glycogen synthase or glycogen phosphorylase. Removing the phosphate, usually by phosphatases, reverses the effect. This way, the cell can quickly respond to its energy needs. Yes, exactly. When CAMP levels rise, it activates protein kinase A, which then phosphorylates glycogen phosphorylase. This phosphorylation activates the enzyme, promoting glycogen breakdown to release glucose. Exactly, there's a balance. When CAMP levels drop, protein phosphatases remove the phosphate groups, deactivating glycogen phosphorylase. This slows glycogen breakdown, ensuring glucose isn't released when it's not needed. Phosphorylase has 2 forms, the active A form and the less active B form. Phosphorylation converts it to the active A form. Glycogen synthase is the opposite. It's active as A and less active as B. Phosphates inactivates glycogen synthase. So phosphorylation has opposite effects on these 2 enzymes. I
to viciously shrimp away from you from here and get the shoulder off the floor and face you. So I need to take a backhand frame, and now from here, I just go in and I lean body weight to my partner's far shoulder. When she goes to start moving out away from me and framing and doing all those things people like, it's not an easy thing to do from here. Now I play a game where I fight to the crossface. I go up first. If she doesn't follow me, I go collar tie, and I pull myself into a crossface. If she follows me, I pommel around and underneath, and we go to a crossface. If she follows me up and down, she's a sticky inside bicep tie, I lift and I drive everything forwards to create a disconnection, and I pommel inside, and from here, we go in and we lock up. We lock up with a 10-finger grip, we play a game right from here. She goes to move around. It's not an easy thing to do at all. And now we open the elbow. We fight to the inside position, like so. We lean forward, and before we do anything, we get that ear-shoulder connection. And she goes to move around from here, pummel the hand back inside. Not gonna happen.